Hi everyone. First, I would like to show the stats of exam 2 and compare the two exams we have taken. It is interesting to see that average from the two exams are basically identical. It's all about the 85.3. But then the curve looks like a little bit more flattened in the exam 2. So this indicated that either you guys have a great performance very consistently, and or maybe my exams are consistent easy, all right? But then this time we have 7 perfect scores, so I'm very proud of you. So please keep working hard, and then after exam 3, I think things will be much easier. Mm. So in today's lecture, we will talk about what can go wrong in our genome and how these changes will affect our health. The genome alterations can occur at different levels. Say this colorful instant noodle sort of structure is chromatin. The changes can happen when long pieces of DNA or chromatin are involved. For instance, we learned the TAD, the topologically associating domain. We know that DNA sequence within a TAD physically interact with each other more frequently than with the sequences outside the TAD, right? So if the things can go wrong, then these interactions during DNA replication and transcription could lead to chromosome rearrangement and gene fusions. We also learned that epigenetic modifications on DNA and histones can alter the chromatin structure and gene transcription. And today we will focus on DNA mutations, all right? So a mutation is a change in a DNA sequence that is rare in a population. Mutations may moderate gene expression and change protein amino acid composition. A mutation may be present in all the cells of an individual or only in some cells. In a germline mutation, the changes occur during the DNA replication before meiosis. So the resulting gamete, also zygote, and all the cells that descended from it will have this mutation. Therefore, these mutations will affect all the cells of an individual, including germ cells. You can imagine that the germline mutations are transmitted to the next generation of the individuals. All right. But in contrast to germline mutations, a somatic mutation happens during DNA replication before mitosis, and is only passed to the daughter cells of this particular cell, but not to all the cells in the individual's body. Therefore, the somatic mutations are not transmitted to the next generation of individuals. So this is a difference. DNA mutations can be classified in another way. There are many ways to classify it, you know. So spontaneous mutation or induced mutations, okay? As its name, the spontaneous mutations happen spontaneously or naturally. These could be a result from errors in a normal cellular processes, such as DNA replication, and we will talk about how this occurs in a while, okay? Spontaneous mutation can also be caused by natural changes in the molecular structure of the bases in DNA. I'll show you some examples. But in contrast to spontaneous mutations, the induced mutations do not occur naturally, okay? Instead, they are induced by mutagens. Radiations those are mutations, and some chemicals are mutagens, okay? The mutagens that can cause cancer are called carcinogens. I'll give you some examples. First, we talk about spontaneous mutations. I just mentioned that spontaneous mutations can be caused by errors in DNA replication. But how does this happen? We all learned that the DNA polymerase uses the sequence of the template strand to add nucleotides to the new strand. And this is always from 5' to 3', right? But DNA polymerase is not perfect. It can make mistakes, okay? In some rare cases, DNA polymerase could incorrectly read the nucleotide on the template and then insert a wrong nucleotide into the new strand of DNA. And this mistake will lead to spontaneous mutations. And the mutation rates vary substantially among different species and even among different parts of the genome in a single organism, okay? So scientists have reported the mutation rates as low as 1 mistake per 100 million to 1 billion nucleotides in certain bacteria. And it can be as high as 1 mistake per 100 to 1,000 nucleotides in a group of error-prone polymerases in humans. You don't have to remember exactly the numbers, but I just want you to understand that polymerase can make mistakes, and this mistake is going to lead to spontaneous mutations. This slide shows another cause of spontaneous mutations. The name showing here is called tautomeric shift, okay? So in figure A showing here, we learned that a normal base pairing between A and T contains these 2 hydrogen bonds, okay? But then in figure B shows a tautomeric shift and produced by a normal and reversible change in T and allow this hydrogen bonding with a non-complementary basis and resulting in T pairing with G, okay? In the next round of replication, the original AT base pair will be replaced by a GC base pair in 1 copy of DNA. Now let's talk about induced mutations. I'll give you some examples of mutagens in our everyday life. So this slide shows the source of radiation that individuals are exposed in the United States, like radon, cosmic, terrestrial, and internal human body are background radiation, which means natural sources of radiation. But the radiation from medical X-rays, like nuclear medicine and the consumer products, are variable from person to person. And then here, radon is the second leading cause of lung cancer in the United States, and you cannot see or smell radon. Okay. So in northern United States, radon is found in high levels in many homes in northern United States, especially when you have a basement. Many home inspectors can do radon testing, similar to what the figure shows. But in Houston, most houses, we do not have basement, so we should not worry about the radon too much. This table shows various sources and doses of radiation we experience during our everyday life. It is interesting to know that we actually receive less than 1% of radiation from a dental X-ray in comparison to like a full body CT scan, which is a much higher of doses. In addition to radiation, chemical mutagens can also cause mutations. For example, the cytosine can be converted into uracil by action of this nitrous acid. This will lead to a base pairing between uracil and adenine. And nitrates and nitrites used in the preservation of meats are converted into nitrous acid in our body. So that's the reason why preserved meats are not very healthy, okay? However, some preserved meat, such as bacon, shown here, is very delicious. So this is a tough decision. This figure shows how a C to U mutation affects DNA sequences. So once this mutation occurs, and then what happened is during the DNA replication, the newly synthesized DNA strand will have a different base here, showing here the GAGTA then becomes GAATA. After 2 rounds of replication, the original this GC base pair showing here is converted into an AT base pair. If a DNA is mutated, depending on how this mutation affects amino acid sequences of the protein, there are several categories. This slide shows several common nucleotide level mutations, right? So this is the normal sequences, and then every 3 nucleotide encodes 1 amino acid. So it gives us this the coding strand, and this is showing all these different amino acids here. So in a silent mutation, the change of 1 DNA base pair, it does not have any effect on the sequence of the amino acids in the protein. So you can see that the normal sequence and the silented and mutated sequence on the amino acid level, they are essentially the same. So both are losing here, okay? Therefore, silent mutation do not affect the structure or function of the protein. So we learned previously, this the third genetic, the third residue of the genetic code. We know that they have a wobbling, right? So even if they are different, they encode the same amino acid. So that's the reason, okay? But then in contrast to that, there's a missense mutation. So this is a change in 1 base pair that results a change of 1 amino acid in the protein. So you see this leucine now becomes a serine because this change is happening in the second nucleotide in the 3 genetic code. And then what is nonsense mutation showing here? So nonsense mutation is a change of 1 DNA base pair and produce a stop codon and permanently signal the cell to stop translating the proteins, okay? So these types of mutation results in a shortened protein that may function improperly or not at all, right? So shown here, TGA, we already learned this is a stop codon, and then if you have a mutation happen to make a stop codon, then no more amino acids will be made anymore, okay, after this stop codon. And a more serious, I mean, this is supposed to be equally serious probably. It's a frame shift mutation. So this frame shift mutation, that means it occurs when the addition or this is the example showing here is a deletion. So either a insertion or a deletion, then these cause a change in gene's reading frame. So we know that the gene actually, the amino acid is made by every single 3 nucleotides, right? So if you have in this case, you lost 1 of the T showing here. So T initials TTACC, right? So now becomes TACC. So you missed a T in here, and then all the reading frame is going to change. So what will happen in this case? So there will change, usually those proteins, the whole amino acid sequence is going to be changed, or sometimes even generate a stop codon. And then usually under this situation, the protein is non-functional, okay? So the frame shift mutation can caused by insertions, deletions, and also sometimes duplications, which is not showing here. So I'll give you an example of how DNA mutation is linked to genetic disease, okay? You probably all heard of this called sickle cell anemia. So in this disease, the shape of a protein's red blood cell look like a sickle. So this, our normal red blood cell is supposed to be a round, very nice looking red blood cell, but then in this anemia, this blood cells, they change into a sickle shape, okay? And then the sickle shaped cells, they carry very low oxygen and can easily stuck in this small blood vessel and prevent the blood flow. So the patients can have anemia, joint pain, and even organ damage, okay? The malfunction of the red blood cells is due to the DNA mutation in this gene called the beta global gene. So beta global gene is located in chromosome 11. In sickle cell anemia, one codon of the T in CTC, the T showing here, mutated to A, okay? So the replacement of this codon is a missense mutation, all right? This is missense mutation. The mutation will change the amino acid sequence, right? And make the hydrophilic glutamic acid become the hydrophobic amino acid called valine, okay? This substitution creates a hydrophobic spot on the outside of the protein that sticks to the hydrophobic region of adjacent hemoglobin molecules beta chain. So this causes clumping of hemoglobin molecules into rigid fibers, then causing this sickling of the red blood cells. Okay, this slide shows how frameshift mutations alter the reading frame and change the amino acids in the protein. The insertion of extra base to genes, showing here. And then this insertion of this adenine changes the reading frame of the mRNA codons, causing a tyrosine becomes a leucine, okay? And also this insertion causes the alteration of all downstream amino acids. So therefore, a frameshift mutation can generate much worse consequence than missense mutations. Previously, we talked about Fragile X syndrome, and several of you had questions on how this happens, okay? So the answer is allelic expansion caused by trinucleotide repeats, okay? So this picture shows a human X chromosome that contains the fragile sites in the long arm. So this site is caused by this allelic expansion of a CGG repeat. Okay, so Fragile X syndrome is one of the most common forms of intellectual disability. It is seen in about 1 in 1,250 male births. So this slide shows the allelic expansion in the FMR1 gene that at this Fragile X allele. This Fragile X locus, okay? So this A showing here is the normal allele. So this gene normally contains about 6 to 52 copies of a CGG, this trinucleotide repeat. But then what happened showing B is the mutated allele, okay? So you can see here, this region, instead of only 6 to 52 repeats, now it becomes expanded into 230 copies of this CGG trinucleotide repeats. So this type of mutation can cause the resulting protein will cause this non-functional or affect the function significantly. And this will cause the Fragile X syndrome disease. Fragile X syndrome is not only disease with expanded trinucleotide repeats. This table actually lists some other genetic disorders with the same molecular alteration. And I believe some of them will be reported in your presentation. Okay, so after we learned so many DNA mutations, you may ask, can DNA mutations be repaired by our own body? If they are not repaired, what will happen? Okay, so the answer is actually yes. Our body is smart enough that can correct many mistakes without you even realizing it. Okay, we have more than 175 DNA repair genes in our genome. And the repair rates were close to damage rates. However, if the DNA damage is too much for our body to fix, the cells will choose to stop growing. That means they go into a dormancy, okay, or die. That means they go to apoptosis. But in rare cases, the unfixed cells can become cancerous. We will talk about that a little later. While you sit here doing nothing, there are actually many DNA damage happening in our body, okay? See this table? These are the rates of DNA damage in a single cell, okay? This is a single cell. If you stand outside an hour during the noontime, particularly in summers, then each of your skin cell will have 50,000 of damages, okay? So stay inside during the noontime, okay? The bottom table lists the maximum DNA repair rates in a cell, okay? You can tell that our body has a very efficient repairing system as long as you don't expose to Texas sun all the time, particularly during summer. The DNA repair system in our body can do many things as listed in this slide. It can proofread during and after DNA replication, repair DNA damage caused by UV. Failure of any components in this system could lead to genetic disorders. We'll talk about one example named xeroderma pigmentosum in a few minutes. This slide shows the proofreading activity of DNA polymerase. During replication, DNA polymerase can proofread the base pairing between the template strand and the new strand. If an incorrect base has been inserted into the new strand, it is removed and replaced with a correct base. Enzymes can also do base pairing substitutions after replication is completed. Showing here is a mismatch of A, C, which can be detected and corrected and then make it back to normal. If the mismatch remains undetected or unfixed, it will eventually become a mutation. So now let's watch a video together that summarizes major DNA repair pathways.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, The enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
A defect in DNA repair can be linked to genetic diseases. For example, the xeroderma pigmentosum is an autosomal recessive disease that has a malfunction in nucleotide excision repair. From the video, we learned that excision repair is needed to fix thymine dimers caused by UV. For instance, it's under the Texas sun in the summer noontime, right? Without excision repair, the thymine dimers remain, and then it will block DNA replication, and the damaged skin cells will die and leave marks on the skin. So therefore, these patients must avoid the sunlight. So this is a picture of a pediatric patient from UK. And then you can see all these dead skin lesions is exposed to the sun. Previously, we mentioned that if the DNA damage cannot be repaired, cancer can occur. Actually, cancer is a genetic disease that can caused by DNA mutations. We actually talked a lot before, right? So this slide lists the numbers of somatic mutations in different types of cancers. So this is by the genomic sequencing of tumors. You can see here the melanoma showing here has the most DNA mutations, all these spots. Average is about 600 coding mutations in each tumor, whereas AML is acute myeloid leukemia. It has less numbers of DNA coding mutations, showing much less, right? Then scientists that have studied the mutations extensively in cancer development. Usually, one single mutation will not be enough to cause cancer, and several mutations are needed. We learned those previously. We know that one mutation, two mutations, you accumulate enough mutation before you really the cancer can be developed. Okay, so for example, we learned in cell cycle, if an oncogene is activated and promote cell growth, the tumor suppressor genes will be activated and try to hold the cells from damage repair or directed to cell apoptosis, okay? But if the tumor suppressor gene also has a mutation and lost its function, then cancer can occur. So that is the reason why most cancers happen in older people, because DNA mutations accumulate with aging. The older you are, the more mutation you have in our body, right? Then why some cancers have more mutations than other cancers? We first learned that the melanoma has a lot more cancers than the AML, right? Is it possible that some of the mutations are more important than others to drive the tumor genesis? Actually, after many years of studies, the answer is a yes. Okay, each cancer is characterized by many somatic mutations. Of those, only a subset of mutations contribute to the tumor's progress, right? We name those critical mutations as driver mutations. Many mutations on oncogenes or tumor suppression genes are driver mutations. Can you give me an example of that? So we learned RB, we learned P53. Those are the driver mutations. Okay, you have those mutations. In contrast, those mutations that do not have impact on tumor genesis are named as passenger mutations, right? The numbers of passenger mutations varies in different cancers because you see, passenger mutation does not seem to have much impact on the biology of cancers. This slide shows the approximate number of driver mutations needed to cause different types of cancer. This was published 3 years ago in the Journal of Cell. Okay, you see that only actually a few driver mutations are needed in various cancer. This is a colon cancer, it needs like 11 driver mutations, kidney cancer maybe 2, and then gastric cancer maybe only 4 driver mutations needed, lung cancer, breast cancer, and brain cancer. They all have less than, this few, less than 10 driver mutations. But then what are the passenger mutations do? Because previously we just mentioned the melanoma averages 600 mutations. Are they useless for this driver mutation? This picture lists the development of mutations in 2 tumor cells. You can see that both cells, they need the same driver mutations to become a cancer cell. This mutation is the driver mutation G, showing the red over here for this different stages development. So meanwhile, during the tumor genesis, many passenger mutations can be generated, and these mutations are different from cell to cell, right? In another word, the set of passenger mutations is unique to each tumor cells. But then why this is important? Okay, because our immune system has never seen these mutations in our body. And our immune cells will recognize these tumor cells by these mutations. Okay, we call these tumor-related mutations as a name as neoantigens. These neoantigens will be presented to the cell surface by the molecule called MHC1. That stands for Major Histocompatibility Complex. We will give a more explanation of MHC molecule when we learn our immune system in the future. Okay, so now you can tell that if our immune system is strong enough, it can recognize these tumor cells with recognize this new antigen and then kill them. All right. However, if the new antigens cannot be very well recognized by our immune system, these tumor cells will survive and further develop into cancer. So neoantigen is the signal that can be recognized by our immune system. Okay, so our T lymphocyte that showing here is our soldier cells in our blood system, immune system. It can bind to this neoantigens through a structure called a T cell receptor, or short abbreviated as TCR. And this interaction can trigger the immune response and then kill this tumor cell. Okay. In the future lectures, we will discuss how scientists take advantage of the neoantigens and design genetic tools to help our immune system to recognize and kill the tumor cells better. Okay. The major point here is that although passenger mutations do not contribute to cancer development, but they are important for T cells to identify and fight those tumor cells. Okay, so for this part, I think most important thing is you need to remember the difference between driver mutations and the passenger mutations, and also understand what's their function in terms of during the cancer development, and also during the process of our immune system to recognize those tumor cells. Right, so that's all for today. I'll see you next time.